My name is Ruthann Rudel, and I'm a toxicologist and a chemist. I study chemical exposures. I'm a research director at Silent Spring Institute, and I'm very excited to be part of the Women Workers Biomonitoring Collaborative, doing really interesting work trying to understand women's exposures in the workplace, particularly for firefighters. And you've heard a couple of the presentations already about some pieces of the work, and now what I'm going to talk about is our efforts to try to identify exposures that we would call unknown unknowns. That is, what are the chemical exposures that we don't know to look for? Can we figure out what they are? That there are some unusual exposures, and can we also use metabolomics to see whether there are biological effects from some of these exposures. I'm going to share my screen. So this is work that has been principally done by Vincent Bessonneau, my colleague at Silent Spring Institute, who is now in France working at a research agency for public health there on their Exposomics and Metabolomics program. But he continues to be involved with this project. And Exposomics is the idea of trying to understand of all the chemical exposures, or many chemical exposures. And metabolomics is trying to understand and measure changes in your biology, your what endogenous molecules in response to exposures. And so we asked are women firefighters in San Francisco exposed to toxic chemicals that might be linked to breast cancer? And are their levels higher than office workers? Our collaborative actually also includes nurses now. We've archived samples from firefighters, nurses, and office workers. We did data collection that included an interview about work habits, as well as some opportunities for exposure to these chemicals outside of the workplace. We collected urine and blood samples. And I want to make sure all the researchers in the group here understand that the samples that from our archive are available for collaboration. We have both individual samples as well as samples that are pooled by occupation, samples from firefighters, nurses, and office workers. We've got a variety of blood sample types as well as urine. We hope you'll contact us if you want to work with these. You've heard about the targeted analysis for flame retardants and for PFAS chemicals from Jessica Trowbridge and Heather before me. And those findings have been published. You can also get those papers on our website. So for the non-targeted analysis, we looked for the chemical exposome. What are chemicals people are exposed to that we didn't target? And biomonitoring is an opportunity to link exposures and health by measuring both chemicals and biological changes, because you can get them both in the blood and the urine. And traditional studies, exposure studies, rely on a targeted analysis of chemicals selected from a priori knowledge. So it's usually a small set of chemicals. For example, we picked, I think, 10 or 12 flame retardants and about the same number of PFAS chemicals. But of course, there's thousands of chemicals out there and thousands that firefighters are exposed to. So this traditional approach of picking a handful of chemicals to look for might miss important exposures. And the non-targeted analysis uses high-resolution mass spectrometry, and it can measure environmental chemicals, which we'll call the chemical exposome, and also endogenous metabolites. That is, endogenous is sort of inside your biology, like your hormones, your growth factors, your vitamins that are in your body, and that would be the metabolome. And the way that the non-target analysis for the chemicals works is that we scan the samples for chemicals using this full scan approach. We have developed a database of chemical compounds, about 1,000 or so, that we thought might be in the samples, and then we look to see whether there are any matches between those. Based on those tentative matches, we select the chemicals we're interested in, and then we validate and measure the individual samples for the chemicals that we chose. In our chemical selection process, so we started with about 600 suspect compounds that matched to our database. We targeted a handful of those, about 20, I think, for confirmation, and confirmed a small number of them using this approach. So our suspects came from many categories of chemicals, included we're interested in nitro compounds, pesticides, perfluorinated compounds, phenolic compounds, phosphate flame retardants, and phthalates. And each of these lines, each of these horizontal lines represents the mix of chemicals, chemical matches that we found in any particular participant in our study within these different classes. So we can see that potentially there are a lot of different chemicals there, and the challenge is, can we identify and measure them? We generally selected chemicals if they were more abundant in firefighters than office workers, if they were not measured by others, if they're expected to be a workplace exposure for firefighters, if they're of toxicological concern, especially for breast cancer, so genotoxic chemicals, estrogenic chemicals, and chemicals that cause mammary gland tumors or alter mammary gland development. And to validate them, we needed to be able to obtain them commercially as an analytical standard. Some of the chemicals that we found using this approach include some that are relatively well known already: benzophenone 3, which is a sunscreen filter; ethylparaben, which is a preservative in lotions and cosmetics. We found an additional PFAS compound that we hadn't targeted in our PFAS analysis, and also an additional organophosphate flame retardant that we hadn't targeted in our flame retardant analysis. We found a hit for Dynaseb, which is a long ago banned pesticide that was used for weeds, and it didn't really make sense that we found it. And then we do have some evidence that it's been used in weight loss supplements, and that might be a possible source of exposure to this compound. And pentachlorophenol as a wood preservative that's been used, like pressure-treated wood on decks or other applications. So the metabolomics part of the work is about connecting chemicals measured through the chemical analysis with endogenous molecules measured in the blood, and to evaluate possible effects on biological processes. So we're looking at building a network of interactions between the chemicals and the endogenous molecules using bioinformatics tools. So they're looking at correlations essentially, but adjusting for many other correlations as we do this. And using this approach, we were able to build some networks of compounds that are known to be related in a network as part of our normal metabolism, and to see some associations. In particular, we found networks of interactions that look connecting parabens with some effects on cholesterol metabolism, and the PFAS with some inflammatory and immune reactions. And those are consistent with some effects reported for those compounds in other studies. So this is a first and exciting and I think first of its kind, application of an approach of using non-target analysis for chemicals plus metabolomics to try to tease apart early biological changes that might be associated with the chemical exposures. So in summary, our non-target analysis identified new chemical exposures that are not currently biomonitored, and that networks of interaction suggest that exposure to PFAS and parabens might affect cholesterol metabolism, inflammation, and steroid hormone biosynthesis in these women firefighters. These are, one is published and the other one is up as a preprint, not peer-reviewed yet, but available if anyone wants to look for more details. And thank you for listening, and thanks to the great team that works on this project. It's a wonderful group, and I hope we'll hear from you. Thank you.